Good morning, everyone. This is our Stanford EMX for December, the last of the calendar year. And of course, we have the holidays coming up, and I'd like to wish everyone safe and happy holidays, restful, amongst other things. And we'll start again for the next year. Hopefully, we're aiming for January the 8th, the first working Monday of the year. But today we have two really exciting speakers again. And so I'm going to hand the baton over to my colleague, Hua Chu, who will introduce the biology speaker. Hua, please.
All right. Good morning, Bob. And let me wait for another minute. So when all the participants have entered the room, I see the numbers are increasing, but I don't know they actually all enter or not yet. So let's give it another minute. Okay, I think they will be creeping in. All right, though, I'm Hua Chu from Stanford Bioengineering Department. It's my great privilege to introduce our speaker today, Dr. Alexis Rohou from the Genentech company in South San Francisco. Alexis now currently is the senior principal scientist in the Department of Structural Biology at Genentech. And he also served as a director of the cryo-EM group. And he have a immense experience in cryo-EM since a graduate student in the Imperial College in London. And his expertise is specialized in software and algorithm development for single particle. And he also had experience in the Harvard Hills Medical Institute, both at Brandeis University and the Genentech Farm research campus of the Harvard Hills Institute. And currently he is in charge of a group that support the drug discovery and basic research across several different therapeutic areas. So we are really excited to hear his experience and his most advanced research in a number of projects, trying to look at drug target using a cryo-EM data for discover small molecule antibody fragments and peptides. And so without further ado, I'd like to give the floor to Alexis to give us the story on his work. Alexis.
Thank you very much, Hua, and thank you, Bob and Hua, for the kind invitation to speak this morning. It's a pleasure to be here. As Hua said, I am from Genentech, which is a biotech and pharma company in California, where I lead the cryo-EM group. This morning, I've got two stories to tell you. The first one is to give you an idea of how structural biology and specifically cryo-EM can help in discovering and designing small molecule drugs against very challenging targets. And in this case, I'll be talking about an ion channel called the voltage-gated sodium channel 1.7. The second story I'll tell you later concerns small proteins. And what I mean by small is significantly smaller than 100 kilodaltons. The first half of the talk will be concerning large proteins, which cryo-EM has been great at doing for many years now. But one of the boundaries of the field is to go towards smaller targets. It turns out many proteins in prokaryotes and eukaryotes are much smaller than 100 kilodaltons. So what can we do to study them? And I'll show you the approaches that we've taken and some success stories with targets in the range of 25 kilodaltons. So first, about drug design using cryo-EM. So I'll be using as an example a project which we published on earlier this year in eLife, and you'll find more details in this paper. And it was led by Mark Schomczak, who at the time was a postdoctoral fellow at Genentech and is now a group leader. And it's looking at NAV 1.7, the voltage-gated sodium channel subtype 1.7. Now, this channel is a very famous target. The reason is that there are mutants in a human population which are very striking in their phenotype. Loss of function mutants of NaV1.7 lead to complete insensitivity to pain. And on the other side of the coin, gain of function mutations lead to extreme pain disorder. And these kinds of data, together with experimental data from the lab, have meant that many people are convinced that NaV1.7 specifically is one of the subtypes of ion channels that is specifically involved in transmitting pain signals up the nerve fiber. And NaV1.7 is a classical pseudo-tetrameric ion channel, in that it's got 4 domains, domains 1, 2, 3, and 4 here in different colors. And each domain consists of 2 groups. First, a bundle of 4 helices, which we call the voltage sensor domain, and then 2 helices that form the pore. And these 4 domains are arranged in a clockwise manner around the ion channel in the center, the pore. The pore modules, the pairs of helices are near the center, and the voltage sensor domains are near the periphery. And as the name implies, these outer domains, these 4 helical bundles, are responsible for sensing changes in the plasma membrane as an action potential is propagating down the nerve fiber. And in response to this change in membrane potential, the voltage sensors have specific helices within them. The 4th helix is very sensitive to voltage, and it moves up and down in response to changes in membrane polarization. And that movement up and down transmits mechanically to the channel, leading it to open and close. And roughly speaking, 3 of the voltage sensors, numbers 1 through 3, are responsible for the very fast opening of the channel in response to the change in membrane polarization. Whereas VSD4, the 4th one, which we'll be talking about some more now, is slightly delayed and leads to the closing and the recycling of the channel, as it were, so that it's ready for the next wave of opening. And in this depiction here, the SD4 in green has a little small molecule in yellow and blue depicted here. And this is the target that I'm going to be talking about. And the idea here is that if we could wedge the exact right small molecule in there to block VSD4 from doing its job, from resetting the channel at the end of the action potential, then the channel would not be available to transmit the next action potential, and thus the pain signal would be intercepted. So for the purpose of this talk, I'll just stay at a very high level on the chemistry that's gone on this target. Because it's a very famous target, many companies have come up with great small molecules targeting it. Roughly speaking, I want to just boil it down to 2 types of compounds. On the left, the aryl sulfonamides, where you have a ring system to the right-hand side of this sulfonamide moiety. And on the right, the acyl sulfonamides with no ring. Okay. These 2 series are quite distinct from each other. They can both be efficacious in some assays, but they have different properties otherwise. One of the big things that every drug discovery program aims to do against NaV1.7 or other targets is to hit only the target and not closely related orthologs. And in this case, there are many subtypes of voltage-gated sodium channels that need to be avoided. So what do we know about how, in detail, these small molecules stop channels from working? Well, one of the big pieces of information came in 2015. After many years of effort and hundreds of protein constructs that were tried and thousands of crystallization trials, one crystal structure was obtained by the group of Jian Payande at Genentech. And it was a structure of one of the aryl sulfonamides. And it was wedged, as I implied before, in the VSD4 domain. And we learned from this structure how these compounds work. They have a so-called polar warhead, where the sulfonamide and aryl moieties are, which interact very tightly with specific side chains on the 4th helix. The one that I mentioned earlier moves up and down in response to voltage changes. They bind to these arginine side chains, and these arginine side chains are positively charged, and they're the ones really that feel, in quotes, the membrane potential and move up and down as a response to it. So these small molecules wedge themselves between 2 of the helices, and then they get stuck against these arginines and they prevent them from moving up and down. That's the mode of action of these molecules. Now, unfortunately, this crystallization experiment was a bit of a one-off. It was essentially irreproducible. We didn't manage to solve any other co-structures of small molecules bound to Nav1.7 using crystallization, despite many, many more trials. And we still had no idea, well, no experimental data that directly told us about how the acyl sulfonamides did their job and how to understand their structure-activity relationships. We did have good models. For example, here is a model based on docking an acyl sulfonamide into the binding pocket of the aryl sulfonamide, so the blue in the pocket of the orange. And, you know, that explains some of the information we had, but not all of it. You had to do a bit of hand-waving to really make everything make sense like this. So really, we needed experimental structures, and cryo-EM was coming along at the time, started to solve structures of ion channels. And so the question was, well, how long would it take us to get to high-resolution structures with these new compounds? And the answer is, it took a very long time. We had to go through many iterations. There were several postdoc projects involved, many, many different trials. So I'll give you a flavor for the sorts of things we tried. At first, we took the same protein that had been used for the crystallization, and we augmented its mass by complexing it with some antibody fragments from some Fabs. So we call these Fab chaperones in the sense that they help chaperone the protein through the structure determination steps. That gave us a few structures. We solved some small molecule structures, but at modest resolutions in the high 3 angstroms, close to 4 angstrom resolution. So not great for understanding drug binding and MOA. 2 talented postdocs, Hui and Thomas, also tried classical things that you can do with channels, like adding insect or spider toxins to them to stabilize them. That led to some success. But really, one of the big differences came when we saw a paper in the literature about using cockroach ion channel rather than human or bacterial or other sources. That expressed better and it behaved better during structure determination. And we're able to chimerize the human voltage sensor domain 4 onto this construct and start solving structures. These were starting to get at good resolutions. And the final step came when after extensive optimization, Christine in the lab managed to get this channel into nanodiscs in a reproducible way and good yields. And then once that key was found, we're able to get reliably high resolutions. To give you the best example, Mark was able to get to about 2.2 angstrom resolution on this target once we had all these things lined up. So what did we learn? Well, we saw a lot of detail in these maps, and I won't show you all of them, but just to give you an idea, when you get to these kinds of resolutions, you see water molecules and you can place them with high confidence. So here is a depiction of all the water molecules Mark was able to place. And that helps with understanding binding modes in detail in some cases. So here is an example of another aryl sulfonamide that we solved by cryo-EM. And you can see this reproduces what I showed you earlier. We kind of knew from crystallography that this would look like that. It does this, what I said before, it engages these arginine side chains. And it stacks very nicely against this particular tyrosine side chain here. Okay, so lots of fine details about the aryl sulfonamides, but we kind of already knew this. With the acyl sulfonamides, we had a big surprise there. They didn't bind at all like the aryl sulfonamides, like we thought they would. Instead, they bound about 90 degrees away from it, and they didn't wedge themselves between S2 and S3 helices, but between these S3 and S4 helices. So this is a binding mode that, as far as I'm aware, at least internally, no one had predicted. The experiment here really told us something new. And when you put them side by side, it becomes obvious that one of the reasons it would have been challenging to predict this is that by wedging itself between S3 and S4, this small molecule creates its own binding pocket, in a sense. In other words, if you took the structure with the aryl sulfonamides, there just isn't the space there to bind between the S3 and S4. And so the protein really is flexible and accommodates different small molecule binding modes. So at the time, a couple of members of the team, and I can't take credit for this, came up with this cool idea. We had several aryl sulfonamide structures. We had an acyl sulfonamide structure. And when you superimpose them in the computer, you could see that both of them kind of met very neatly in the same area near the warheads. So what if you could hybridize these two things and create a chimeric, not so small molecule anymore? Would that work? The team tried, and the first compound they designed actually wasn't too terrible. It went from the parent compounds were in the low nanomolar potencies. This was about 50 nanomolar potency, not too bad. Mark solved the structure of this, and here it is. You can see that the design worked. We obtained a compound that bound using both of these binding pockets and merged them together. It wasn't perfectly relaxed in this pose. The stacking against this particularly tyrosine that I pointed out earlier wasn't quite right. But with a further redesign from our chemistry colleagues, simplifying the right-hand side of this chimeric or hybrid molecule, we got back to 9 nanomolar, solved the structure by cryo-EM, and now this looks like a much more relaxed pose with beautiful stacking of the left-hand side of our chimera with this tyrosine. And so this was an example of pretty extreme redesign of small molecule compounds, really that couldn't have been imagined without cryo-EM structures supporting the effort. And if you've ever seen sort of structure-based drug design talks before, this is quite unusual because normally you grow what you have step by step, and you increase and improve the small molecule. In this case, when the team tried step by step, going from an aryl sulfonamide and growing progressively towards the acyl pose, those compounds didn't work. You really needed to do the merging of both and go all the way from the membrane to the other side of the voltage sensor domain for this to work. Okay, so this is an example of where extensive work over multiple years to engineer protein, together with a lot of biochemistry and together with lots of great software and state-of-the-art microscope, led us to be able to solve structures quite routinely almost of a very challenging target and to improve our drug discovery capabilities. So the next story I want to tell you, as I mentioned before, is a more technology story and trying to talk about how we approach the problem of small proteins. Now, in cryo-EM, we don't have a very good track record as a community solving small protein structures. And there's a fundamental reason for that. Small proteins are unlikely to have low frequency features that you can easily use to register images with each other, especially in a low signal to noise ratio situation like we have in cryo-EM. The high frequencies that you need to align small proteins are masked by high levels of noise. And in fact, there's a theoretical fundamental limit to how low you can go in molecular weight, probably around 20 or 30 kilodaltons. We're not sure exactly. But it's very challenging to solve structures of proteins in the 50 kilodalton range, although it can be done. But to make things a bit more routine, many groups have been trying to, well, of course, improve the instruments, improve the software and so on. But you can cheat your way out of this problem, probably, if you can find binders to your target that don't affect its function, but just make it larger so that you can look at it more easily with cryo-EM. And one of my favorite solutions to this problem is out of the lab of Todd Yates at UCLA. What Todd's lab has managed to do over the years is design incredibly rigid protein nanocages, which are decorated by binders, in this case, proteins called DARPins, which can be designed to bind or selected to bind any target of interest almost. And so in this example from this recent paper in PNAS, Todd's group bound KRAS, a famous oncogenic kinase, to their DARPins and their cages. And by doing this, they were able to solve pretty high resolution, about 3 angstrom structures of KRAS. And this is beautiful because it gives you symmetry, it gives you a lot of mass. This is an easy object to work with in cryo-EM. However, in-house, we don't have capabilities really to screen for DARPins. And so we're interested in this technology, but just watching it at this stage. Another thing you can do for example, is you might already have a binder to your protein, like a nanobody, for example, a special kind of antibody from organisms that make very, very small format antibodies. And in this paper, these authors described a method whereby nanobodies, which by themselves are too small, can be augmented so that the nanobody binds to your target, and then you can add a Fab that binds, a Fab being an antibody fragment from IgG, that binds to your nanobody, and another protein that binds there. And they had this concept of that they call Lego bodies, where you, like Lego bricks, you can build up the mass on top of your nanobody. And they showed some success. They could solve their targets in the sort of 3 to 4 angstrom range resolution. Now, we at Genentech really like antibodies, IgGs, and the fragments from them, the Fabs. We can use them for crystallization. We have, and there are commercial available antibodies and Fabs against many, many targets. We have many colleagues who are really talented at discovering Fabs against any target that we can come up with. They also have the advantage that they add 50 kilodaltons in one pop, and compared to the nanobodies, that's quite a good feature, because let's say you had a target around 10 or 20 kilodaltons, then you add a Fab, you're at 60, 70 kilodaltons, challenging but not impossible by cryo-EM. And you know, they have a very distinct shape, so even in the low frequencies, you can recognize the orientation of a Fab. So great, they sound great, but you can't use them really for high resolution work normally, because they're intrinsically flexible. Fabs can be thought of as 2 domains hooked together by flexible hinges. The variable domain, which has all the hypervariable loops that bind the target and recognize it, and then the constant domain, which hooks it up to the rest of the IgG. And the elbow, the hinges between those 2 domains are flexible. And so if you look at all the PDBs that have been deposited of Fab structures in the public data banks, there's a very wide range of angles. These things are just flexible. Here is just one illustration of this in cryo-EM. A few years ago, we published a study looking at a membrane protein called CD20 that has 2 Fabs bound to it. And you can see that the constant domains of the Fabs are kind of smeared out, because when we average thousands and thousands of images in single particle cryo-EM, we get the average, and the average of something that's moving is very blurry. And so this is a problem. It means we can't use Fabs like this for solving small protein structures because they're not rigid enough. Jennifer Kung, who was a postdoc fellow at Genentech until very recently, and her supervisor Sudhamsu, who is a group leader in our department, a few years ago started to tackle this problem. And they were not the first, but I'll hope to convince you that they were really successful. They tried to engineer Fabs in a general manner to make them rigid. So Jennifer's workflow started by designing mutations with Sudhamsu, expressing the protein, purifying it along with the target, checking that the mutations didn't mess with the binding, and making cryo-EM grids and solving the structures. And she and Sudhamsu went through this process many, many times, tried many, many different constructs until they arrived at a general solution. The goal here is to solve small protein target structures, but here we're going to start by studying the rigidification process by using a much easier protein. We have this extracellular protein, which is a tetramer, and we have 4 Fabs bound to it. So this is a complex of about 300 kilodaltons, very easy to do by cryo-EM. We already had it by cryo-EM in-house using wild-type Fabs. And so the idea was, let's test these mutations to see if they really rigidify the Fabs. So the first iteration, version 1 of the rigid Fabs, you can see here that the binding affinity was preserved. The mutants don't mess with the binding. And Jennifer was able to solve a structure of these first version rigidified Fabs to just below 3 angstroms of this big complex. And this is the structure, the wild type structure with local resolution colored in. So blue is more stable, doesn't move as much, higher resolution. Red and white, lower resolution, more flexibility. That's the wild type structure on the left. And with the first iteration at mutants, it did seem that there was an increase in resolution, which was consistent with rigidification, perhaps not very striking yet. Jennifer and Sudhamsu then iterated, starting from this, added more mutations on top of what they'd already introduced. And gradually, over several iterations, by adding more and more mutations, in the same system, in a controlled experiment with the same number of particles, same instruments, etc., we're able to achieve significantly improved resolutions. Again, consistent with first, the fabs having been rigidified because we could resolve the constant domains a little bit better every time. And secondly, this rigidification helping with resolution. So here is an illustration of these structures through the different mutation ring experiments. And you can see that the more you go to the right, the more fine detail you can see. For example, these water molecules begin to be well resolved from generation 2 of our mutants. Here's another example. This is a tetrameric membrane proteins bound by 2 fabs. And with rigidification, the fabs become better resolved. In this case, this particular membrane protein has a lot of intrinsic flexibility, so not a super striking result. But it shows that these designs, these same mutations can be applied to different fabs. Here is this another membrane protein I actually showed you earlier already, 2 fabs bound to it. And here you see a striking improvement in the quality of the reconstruction. These fabs really were rigidified. But I also want to take a little detour for 1 minute to again make the point that these relatively large proteins are not what we're primarily targeting here with these rigid fabs. In fact, when you have large proteins, they're likely to have their own flexibility intrinsically, and so binding a rigid fab to them may not be the best thing you could do. So what do I mean by that? It turns out that when we bind rigid fabs to this membrane protein, the fabs are so rigid, they're actually more rigid than the protein itself. And so during the alignment of the images in single particle cryo-EM, actually the fab region dominates the alignment at the expense of the target protein. If you look at the bottom here of the protein, it's less well resolved than the parts in the fab, which is not really what we want. Now, computationally, we can overcome this. We can do local refinements where we exclude the constant parts of the fabs, or even local refinements where we just consider the membrane protein. And when we do that, we do recover the bottom of the membrane protein. So we're having to fight against flexibility in different parts of the system here. But by combining all these results together into a single map, using a neat tool from Phoenix, we obtain the map that I showed you in the previous slide. Anyway, all that to say that the rigid fabs are rigid, and they do help, but you have to be careful what you wish for with larger proteins that have intrinsic flexibility. Really, the killer application here is for small proteins. So let me show you some small proteins. So the first one I will show you is 26 kilodaltons. This is Jennifer's summary of the purification. This is a protein we already had fabs against. She took one of the wild type fabs and applied the same mutations as before. And then with pretty straightforward image processing, arrived at about 2.7 angstroms of this structure. So here is the protein on top, this small protein 26 kilodaltons, and the fab. And you'll notice here, we don't have any of these intrinsic flexibility problems. The protein is so small that it doesn't even, the flexibility it does have doesn't make it move very long distances. All parts of the fab are well resolved, including the constant domain. If I color by local resolution, you'll see that the constant domain still is slightly worse than the rest of the structure, but everything is, better. Almost everything is better than 3.2 angstroms probably, and the target certainly is well resolved. So here are some detailed views of the target in pink, and then the light and heavy chains in yellow and gray. So this rigid fab, unlike a wild type fab, really does allow you to solve structures of small proteins. Here's another example. This is a 21 kilodalton protein. And again, no need for special image processing here. Pretty straightforward workflow to get to about 2.85 angstroms. And this is a small protein that happens to have 2 small molecules bound to it, here highlighted in pink and green. Again, local resolution suggests that the resolution is pretty homogeneous, not perfect, but pretty good by cryo-EM standards. And certainly, just to mention here, you still have the choice of playing tricks. You can do local refinement by excluding the constant domain. Numerically, in this case, it seems the resolution has improved a little bit to 2.75. Likely that doesn't make a very big difference. Either way, the small molecules and the protein are well resolved. Again, this is a 21 kilodalton target with just a single fab that's been mutated using these rigidified mutations. So That's the approach we're taking right now to target small proteins with cryo-EM, especially when they can't be crystallized. And this paper is about to be submitted, and all the details for the mutations will be publicly disclosed shortly. So that's it for me this morning. I'm happy to take any questions. Thank you.
Good, Alexis. Thank you very much for these excellent presentations to give us exposures how you tackle these really important and pharmaceutical problems. Let me see if there are any questions from the floor. And let's if you have any questions, please type in the Q&A.
Looks like there are a couple in chat right now. Well,
I can see the Q&A. Would you like me to try to answer them directly?
Right,
Juan?
I wanted to do that, please.
Yeah, so the first anonymous question is, what is your opinion on your new blush regularization in Relion 5 that improves processing with on data with low SNR? Do you think it potentially eliminate the need for large binder proteins to resolve small molecules in the future, small proteins in the future? I think yes, it depends what you mean by small protein. I think regularization techniques like this push the boundary. If before your limit was 100 kilodalton, maybe it will make the new limit will be 80 or 90. So certainly it helps push the limit. In my opinion, there's always going to be a lower boundary to what you can solve, even with the best image processing software. And then at that point, you might still want to resort to binders. Next question. What is the resolution of the cryo-EM structure that would have sufficient information to guide the compound optimization, such as improving stacking in this case? My rule of thumb is from about 3.5 angstroms, things get interesting. Of course, it depends on the shape of the small molecule, but many small molecules that are drug-like, at 3.5 angstroms, you'll have most of the time an unambiguous idea of how it fits in the pocket, where the pocket is and how it fits. And usually a good computational chemist can take you the rest of the way. Now, that is not ideal. We always try and go to more like 2.5 angstroms, and there you can start to see water molecules. You have no ambiguity about the side chain orientations and so on. So it really depends where you are. If it's the first time you're going to see a binding pocket, then 3.5 is going to be enough, and then you can optimize and get better and get higher resolution. If you're really doing structure-based drug design and trying to do fine adjustments, then you want to be in the sort of 2.2 to 2.8 range, I would say. And then, are mutations for rigidity applied to all fabs? Yeah, so in this case, we're able to use the same mutations against all the fabs that I showed. Now, you would have fabs can come from different frameworks of IgGs, and depending on those, you may have to make adjustments. But within a given framework of IgGs, it's very predictable where we need to make these mutations. And almost all of the time, the mutated fabs express well, but that's something that we occasionally still have to give up on a particular mutant because for a reason we don't understand, it doesn't express well. So the answer is yes, almost all the time, the same mutants. And then, Eva, hi Eva, thanks for the question. When pushing the structure of a human channel that has many isoforms that you have to distinguish, does a non-human, was it Scorpion, tell you anything about how it distinguishes human isoforms? So the toxins, let's see. I guess it depends. Classically, the toxins have been used in years past to distinguish different isoforms or subtypes of channels. And some insects have evolved toxins that will hit human channels. So you can, in some cases, use toxins directly on human channels, and that can help you distinguish the subtypes. I hope that answers the question, Zoe. I'm happy to follow up later if we need to. Well, should I keep going with the questions? Do we still have time, or?
Yeah, keep going. I think you are answering well.
Okay. All right. Doesn't adding the fab to the small molecules create a problem with the natural orientation of the molecules? Are the resolution homogeneous? Very good question. This is a question from Cyan Bacta. The honest answer is yes. We often see that fabs, like other proteins, will have preferred orientations in the ice. We have had some luck with detergent additives that people use for proteins in general to solve preferred orientation. I can't claim that we have 100% success rate. This is like the rest of cryo-EM, still an experimental field where you have to try different tricks and solve the preferred orientation problem. I'll say that the small proteins themselves also are likely to have preferred orientation. So you've just kicked the preferred orientation can down the road by adding a fab. You haven't solved the problem, but at least you've got your size in a range that makes it easier. That would be my answer. Jane Richardson, thank you for your question. Really great. How often does this cross the important limit of 2.5 to see backbone conformation? We crossed the 2.5 angstrom barrier in general in a minority of our projects, but a growing minority of our projects. In fact, this year internally, we've had a few projects that went below 2 angstroms on real target. But I think you're probably asking specifically about the fabs and augmenting the mass of the small protein targets. The 2 examples I showed you are fairly representative. We've been able to get to 2.5 angstroms, once or twice. It's, on the examples that I showed, we couldn't really unambiguously see the backbone conformation directly. So I'd say it's still a little bit too rare for our liking. And why? It looks like you want to answer this question live, or?
No, keep going. Yeah, I think I agree with your response.
Oh, okay.
But I think there are now perhaps some other new software. It's a kind of AI-based software can resolve some of the less resolved part better. But I think there's progress. Yes.
I feel like, Jane, that it would be nice if we could see the backbone carbonyl conformations more often and just get rid of that ambiguity. We're just not quite there yet, but all these things coming together hopefully will help. Tillman, hello Tillman, long time no see. Have you tried to stabilize the fab fragments with a nanobody to stabilize? Works also quite well. Yes, I agree. We did try that. We did not just try that. That was like our workhorse solution until we figured out these mutations. So Tillman is referring to nanobodies that specifically target the hinge of IgG fabs, and they will thus reduce the conformational flexibility of the fab. And yeah, that's a great solution. Absolutely works. We've now, since we've figured out these mutations, we've been using them over and over again. Haven't really had to rely on these nanobodies for a while, but definitely a solution that works. And anonymous attendee, how can you accelerate the process of screening gains in cryo-EM? Do you always need to resolve the structure by cryo-EM, or are there other methods to screen conditions such as negative staining? I suppose I'm not exactly sure what you mean by screening gains. If we're talking about, say, you change a small molecule and you want to check whether it's gained in binding affinity and so on, you may have binding assays in some projects, although not all proteins can you do an SPR with. There are, but in general, if you're talking about gains in resolution, then I think you do have to do a structure. You have to do a full experiment, get to the final result. So I hope I got that about right. Eva, also using cryo-EM as a screen for rigidity of fab seems an overkill, not very fast. Was there no other way to screen mutants? There may have been. I'm not sure how to answer that one. There may have been, and I'd be glad to hear about how we could have run this project more efficiently. But in the end, we got the information we needed. Thanks.
But I think, don't you think now the solving a structure is pretty quick now? So the time involved is actually not a limiting factor.
Yeah, although, it's still many hours, right? Like even in the best case scenario, it's still many hours. So I guess if Eva has a biophysical tool that can be done in just minutes to check the rigidity of a fab, I agree that might be even better. I just don't know what that would be.
What sort of dose rates are you using to get 2.5 angstroms, to put it in the context of for material science people?
Sure. So we measure the dose rates in electrons per unit area. So I'm not sure if you may have to convert this a little bit, but something like on the order of 10 electrons per square angstroms. The order of magnitude.
Yeah. Okay, that's
pretty low dose, right?
very beam sensitive materials for us, and it's about similar. Yeah.
Okay. Yeah. Marin, great to hear from you. Thanks for tuning in. What pixel size are we using for 2.7? I'm going to go out of a limb. I think that was our standard pixel size of 0.8 angstroms. So even by your standards, we were okay within a third of or 3 times the pixel size. But thanks for checking us, Marin. And Gwendalia Marini, could you give any suggestions on dataset acquisition for small molecules? Faceplate, gold grids, and would 200 kV cryomicroscope be suitable somehow? So I'll answer the last one first. 200 kV, I think, should be suitable. I think there are examples of 200 kV instruments being used to solve small protein targets, no problem. You just have to align it very well, treat it like a Krios. And the other question is very important. The detector is probably the second most important thing. You need a very high quality detector that's going to give you high signal to noise ratio into the high frequencies. Gold grids, yes. Pick the grid type that reduces beam-induced motion as much as possible. Faceplate, no, not with the current technology. I think we have to wait for the laser faceplate to come out before I would recommend faceplates for high-resolution single particle work. The exception here would be if you have a fairly small target and you're interested in measuring conformation without solving high-resolution structures, but just visualizing conformational heterogeneity or compositional heterogeneity, then the faceplate may make sense. And I think Eva Nogales' lab has got beautiful results that point in that direction. But for high resolutions, I would probably advise against faceplate at the moment. So I hope that helps. Did the hybrid protein with human cockroach become amenable to crystallography? I don't know is the honest answer. I cannot recall whether we did crystallization trials on those chimeras. And from your talk, I had the impression that long flexible proteins are still the most challenging targets for high-resolution cryo-EM. What are the most effective strategies for those? Yes, long flexible proteins are still very, very challenging. Anything that has intrinsic flexibility is a challenge. Now, I don't know how long you mean by long. The longest ones are the filaments, that can be flexible. And here you can take advantage of what you know about the geometry of the object to use as restraints during image processing. And in general, I think for things that are very flexible, where the signal to noise in the data would lead to overfitting because you have too many parameters to fit both the orientation, location, as well as the deformation, then you're going to have good priors on what the deformations are like. And if you have good prior information or a concept or a model of how the deformations work, then there are more and more software packages that can use that information to help you with the refinement. So this is a very vague answer. I'm sorry, this is a big topic. And it looks like we reached the last question. Does increased magnification help with small proteins, e.g. going from 105K to 130K on Krios? The answer is it should help in theory. If you look at the DQE curve of your typical camera, it does decrease quite fast in the middle range. And so if you move to a higher magnification, smaller pixel size, then the same part of the DQE spectrum will now cover high resolution information. So you should have improved signal to noise ratio. And so that's one of the things to try. You then just have to make sure this higher mag is really well aligned, etc. And in practice, it rarely makes the key difference. But I think it should make a difference in the right direction. And with small proteins, if you get your concentration high, smaller fields of view is really not a problem. You should have many, many, many proteins in a small field of view. So I think it's a sensible thing to try. And then as well, Marin will not be able to ask you about the pixel size relative to the final resolution because you'll have a very small pixel size.
Okay, real good. All right, I think that concludes all the questions being raised from your excellent presentation, Alexis. And we thank you very much. Your talk definitely generated lots of interest from the audience by the number of questions being raised, and we thank you.
I appreciate it. Thank you.
I get back to the floor to Bob.
Okay, thanks. Yes, thank you very much, Alex, and thank you for answering those questions so efficiently in the time available. It's really appreciated. I'd really like to then move on to the physical science speaker, and I'm going to introduce Jen Dion to give the formal introduction. I'd just like to say that I'm very pleased to see that Dr. Erdmann Spiecker from Germany is giving our talk today. I've followed his career and his talks for 15, 20 years now. I'm always really excited when he comes on. He has very thoughtful materials, science experiments, wonderful electron microscopy, and this talk was something he gave in Salve a couple of months ago, also in Germany. And it's really astounding what you can do in situ in an electron microscope these days. So Jen, thank you for wanting to introduce Erdmann, and I'll pass it over to you.
Great. Thank you, Bob. I think your introduction said it perfectly. Erdmann, we haven't had the pleasure of meeting in person, but I'm really excited to hear your presentation. Professor Erdmann Spiecker is a full professor at the Friedrich Alexander University in Erlangen, Germany, where I imagine it's probably quite cold but beautiful this time of year. He is affiliated with the Department of Material Science and Engineering, where he also was previously head of the department, and he's also affiliated with the Institute of Micro and Nanostructure Research. In addition, he is chairman of the Center for Nanoanalysis and Electron Microscopy. And just to name a few of his really impressive research accomplishments, he's an expert in situ scanning transmission electron microscopy, having made significant developments in the field, including direct manipulation of individual defects in two-dimensional materials and in nanomaterials in situ. And more recently, he's also extended some of this work to high-resolution X-ray microscopy, so that way he can correlate some of his in situ TEM work with three-dimensional analysis. So without further ado, Erdmann, the floor is yours.
Yeah, thank you very much, Jen, and thank you very much, Bob, for the kind introduction and also for the invitation. It's a pleasure to be here today. Let me share my screen. So is it now correct?
Yes, perfect.
Okay, so thank you very much again, and it's a pleasure to be here. I want to talk about twist-enabled superlubric sliding in bilayer graphene. So I want to talk about a very fundamental experiment that I dreamed of for many years, and you see it here on the bottom already. We wanted to make a real shearing experiment with bilayer graphene, so like in a textbook, to see how the shearing works and also have the opportunity to see the defects moving, which make the boundary. And in particular, what we experienced is that when twist occurs, then we can reach a superlubric regime, so regime of very low friction. And this was possible because of my excellent postdoc, Shou, who made really dedicated work and finally achieved this experiment. And he was supported by Mingyang Wu, my senior scientist in electron microscopy. So if you shear bilayer graphene as shown here, and you just make a simple sketch of a rigid graphene and bilayer graphene, if we would move like that, as shown here, let me start it. Okay, would see that the structure goes out of registry and in registry again, if we just assume that it's a rigid graphene layer which slides on top of the other. However, if you have a twist angle, then you get a kind of moire pattern, and there's a lot of interest in these moire patterns nowadays. And if we now twist, you see, if we now slide the two graphene layers on top of each other, we see that the pattern actually remains. So the pattern is always the same, only the pattern moves a little bit perpendicular to the sliding direction. And this is actually the concept of structural superlubricity, which is nicely illustrated here in the Nature paper by Hott in 2018. So if you have an egg box like here, and you are in registry, then it's very difficult to slide the one egg box on top of the other. However, if you twist the egg boxes, then they do not lock in, and you can achieve a sliding which is much less frictional. Before I come to this current work that I want to present, I want to a little bit introduce how we entered the field of dislocations in graphene and defects in graphene. So we started about 10 years ago and studied in some detail dislocations in bilayer graphene back then. And there was a lot of interest also from other groups in this topic. Then later on, we achieved in situ manipulation of dislocations, so we could manipulate individual dislocations by in situ microscopy in the TM and SEM. And later on in this context, we also developed a cleaning mechanism which allowed us to clean graphene to atomic cleanliness by in situ mechanical methods. And we used this later on to image individual molecules covalently bound to graphene. I want to show a little bit about this early work. We started back then investigating graphene on silicon carbide. So you can grow graphene on silicon carbide by removing the silicon so that the carbon remains on the surface and forms a graphene under certain conditions. And it was, of course, extremely difficult to make these plan view TM images because you have to finally remove the silicon carbide from the back. And this can only be done by etching and laser-assisted etching, and you have to keep the graphene intact. But finally, it worked out, as you see here from the AFM image and the TM image. And what we realized, there's it's full of defects, which was not expected. And later on, we worked together with our colleagues in physics, and they found a linear magnetoresistance, which was not expected, and which could finally be explained by these dislocations. This was work when still Benjamin Butz was in my group, and we had a PhD student, Christian Dolle, working on this topic, and we worked together with Heiko Weber from our physics department. So there are some peculiarities of dislocations in 2D materials or graphene. If you have a dislocation in the bulk material, you have, of course, a strain field like shown here for an edge dislocation. However, if you make the sample thinner and thinner, it starts to buckle in order to release part of the strain energy. And this has been studied many decades ago already by the Ammeling's group. There's a nice paper from Siemens in 1962, and he derived a formula which is very similar to a small angle grain boundary formula, saying that the buckling angle is proportional to the Borges vector divided by the thickness of the film. If we now go to an extreme, and this is just an illustration of a bilayer graphene, and we assume that the thickness is only 3 angstrom or 3.4 angstrom, we would expect a buckling of 20 degrees. So a huge buckling because the sample is so thin. In fact, this is also what we observed and what we could show experimentally back then. But here I want to, just for illustration, show the MD simulation of my colleague Bernd Meyer. And you see here in a bird's eye view, the graphene with these dislocation, and at each edge dislocation, you have a buckle. And the side view show that we actually have a buckling angle of 17 degrees, which is very close to what we would expect from this early formula. There's also a difference if you change direction with such a dislocation, and here's an image where it was here more or less a screw dislocation. Here's a Borges vector, and then it becomes an edge dislocation. The width of the dislocation contrast increases dramatically because of this relaxation. So the strain is dispersed, and so the contrast in dark field imaging also expands. And similarly, this has also been observed by Alden and co-workers, but here it was supported on other graphene layers, so it was not the buckling. Already from the elastic properties, you expect that the edge dislocation is a little bit wider than the screw dislocation. I want to comment very briefly on the nomenclature, because from different fields, people worked on these dislocations in bilayer graphene, and they use different wording. So some called it domain walls, others stacking boundaries, because the stacking changes from AB to BA. And then the term strain soliton was created. And there's a tensile strain soliton and a shear strain soliton. But finally, from material science point of view, this is a partial dislocation. So there's one peculiarity of dislocations in bilayer graphene, that all the dislocations are partial dislocations, which means that the Borges vector is not a lattice translation. So you make a stacking fold, because the stacking fold has no energy, because AB and BA is equivalent. There's no need to make a perfect dislocation. So all this together can be described by the concept of dislocations, and in particular partial dislocations. So based on that, we had the idea to manipulate dislocations, because if you have a dislocation confined in such a thin film, it might be possible to manipulate them mechanically. And this is very nice work carried out by Peter Schweitzer, former PhD student, together with Christian Dolle. Peter Schweitzer is now at NSEM, and also Peter Denninger helped with the setup for the SEM that we used back then. In order to do that, we also developed a cleaning approach, and I show you here a video where we clean graphene with a manipulator from one side and then from the other side. And we could also do that as well in the TEM, and could check that we really get atomically clean graphene or bilayer graphene. In this particular case, it's bilayer graphene, and you see already a contrast line on the top side now in a second. And these are dislocations that were displaced by the tip. And based on that, we had the idea to now manipulate dislocation with the tip. And this is shown here, so we can draw dislocations individually. You see interesting anchor points of these dislocations. Maybe I start it again. Wait a second. So you see anchor points. These are threading dislocations where the dislocation go through one of the top or bottom layer, but you can really explore now the properties of dislocation, like in this particular case, the line tension. And we extended our approach by introducing a third manipulator, 2 for cleaning and then 1 for manipulation. And then you can play with the dislocations with a guitar. So these are dislocations which are anchored at so-called threading dislocations, and you can individually draw them across the graphene. And you can analyze all that in detail, and we could show that actually the anchor points which are sessile are these threading dislocations penetrating to one layer, and there it's a normal dislocation in monolayer graphene. I just want to mention that there are other means to image dislocation, and there's a very nice work by Jiang and co-workers who used a plasmon reflection to reveal dislocations on a bulk substrate. And they could also manipulate these dislocations, but it's of course not possible to image the dislocation while you manipulate them, but you can only do this afterwards. But it's a very nice technique to do that as well. Now I'll come back to the experiment I want to talk about, which is also about dislocations, because a twist dislocation network is also composed of dislocations in bilayer graphene. And I just want to mention briefly that structural superlubricity originated from a paper by Aubry in 1983, and he proposed that under certain conditions there's a transition to superlubricity, so friction-less sliding, if you have an incommensurate structure between the two parts that slide on top of each other. And this was nicely demonstrated experimentally by Dean Weber in a famous experiment on graphite. And I want to show this briefly. So he moved a graphite flake on top of graphite, and then he assumed that the graphite flake was attached to the tip, and he could then rotate the graphite below and measured the sliding friction force with a special Tribo lever as a function of twist angle. And you see here the lock-in at 0 degree and 60 degree, where you have strong friction forces, and in between you have very low friction due to the incommensurate or quasi-incommensurate structure. There's another very nice work where monolayer graphene flakes were moved with the AFM as well on top of a graphene, and they observed when the graphene flake rotates from the locked-in position or orientation, then the sliding friction was reduced considerably, but then after a while it was locked in again due to thermal fluctuations. And our idea was not now to do a similar experiment, but in the TM to see what's going on at the interface and what type of defects are involved. So I come now to the experiment, and it works like that. So we made a dog bone sample for a conventional straining holder, and with a laser cutter we made some cuts and put some graphene, CVD graphene on top. If you now pull on the graphene, and it's a bilayer graphene and partly monolayer graphene, if you're lucky, you can achieve a situation as shown on the left-hand side. And it's really very difficult to obtain such a sample, but finally you find these sample regions where you get this configuration. So you see here a bilayer graphene stripe, and on the left and on the right you see monolayer graphene, and obviously the bilayer graphene has been disconnected from the bilayer graphene on the left side and slided already a little bit to the right. So we have exactly the situation that is shown on the bottom, on the top right, that one monolayer graphene, let's say the top monolayer graphene is on the right, and on the left it's the bottom monolayer graphene, and in between we have the overlapping graphene layers, which make the bilayer graphene region. And if we look a little bit more carefully, we see already that there are dislocations, so lines. This is a dark field image formed with a 1:1:10 reflection, and you nicely see these dislocations at the interface of the bilayer graphene. If we now pull on this dog bone sample, we see that new dislocations evolve here from the bottom part and move up, and you get a network of dislocation which becomes more and more dense the more we pull. And of course the overlapping region is reduced, and finally the two monolayer flakes are separated from each other. So let me go back. I want to say a few things. First of all, you cannot see all the dislocation at the same time, because in one of the dark field images you see only two or three types of these dislocations. There's the third dislocation type which you cannot see, so you need more than one dark field image. So therefore the network here, for example, looks like a little bit asymmetric. There's another dislocation line family which is not visible here. Okay, now of course we see that a dislocation network evolved, but we want to learn more about the process. We think that twisting sets in, but we have to prove that, and for that we have to make a bogus vector analysis. And this is shown now on the next slides. So here's a summary again. In the beginning we have some arbitrary dislocations with different bogus vector, but after first steps of sliding, a network evolves, and this network is made of, as we will see in a second, of screw dislocations. So the almost pure twist boundary. As I mentioned before, in order to completely evaluate such a network, we have to combine 3 types of, or at least 2 dark field images, and then we can derive the full set of dislocation lines and Borges vectors. And here it's enlarged, and you see the 3 types of Borges vectors. And actually all dislocations here, the green is parallel to the green arrow, the red parallel to red and blue, and so on. So it's almost exclusively made of screw dislocations. Okay, now how can we evaluate the twist angle? This is pretty straightforward. There are 2 ways to do that. Either we just rely on the density and the mesh size of the dislocation network, or the moray, so to speak, or we evaluate with 4D stem on the left and on the right side in the monolayer graphene region, the relative orientation of the diffraction patterns. And actually, this is shown here, where we measure on the left and on the right of, for example, this area here, and we see that there is a rotational shift of the diffraction spot here, which we can measure and determine the twist angle locally. So you see that the twist angle changes from bottom to top, because also the mesh size changes from bottom to top. So if we do that and apply these 2 techniques, we can determine the twist angle as a function of position from bottom left to top right, and as a function of the width. So for each step of sliding, we evaluate the network, and you saw that with increasing sliding, so to speak, the twist angle also increased. And I want to mention one thing. It would be nice to have a video of that sliding and to see the dislocation actually moving, but this is not possible because it's a stick slip motion. So if you pull on that, then suddenly there's a jump and it slides for a certain distance, then it arrests, and then you have to pull again, and then, like in a stick slip motion, then it again slips. So we can only do interrupted sliding, so to speak. But you see now the twist angle is very consistent, independent of whether you measure it with 4D stem or with a mesh size from the moray. Now, maybe even more important, because the twist angle is pretty easy to measure, is how can we measure shear forces? And the idea here was that we use a monolayer graphene as a kind of spring. So we know the elastic constant of monolayer graphene, and we know that we pull on monolayer graphene on one side and on the other monolayer graphene on the other side. So by measuring the strain in the monolayer graphene and using elastic constant, we should be able to measure the force or the stress, the shear stress in the bilayer graphene. And this is shown here on the right, how we achieve that. Oops, now I was too fast. It goes back. Okay, here on the right, you see we measure the diffraction pattern with 4D stem at each position here, shown exemplarily for the red dot. And we compared it with a reference diffraction pattern, which is very close to an edge where there's no strain expected because of the free surface. And you see that there's not only a rotation involved, but there's also an epsilon, a strain, which indicates a straining along the strain direction. If we now evaluate both the twist angle on the left shown and the strain in the monolayer graphene, then we can see that they are correlated. So with increasing twist angle, and this is the result for 2 different samples, one I showed here and the other is an independent experiment, we see that in both cases with increasing twist angle, the strain that we measure in the monolayer graphene decreases. And this indicates that the kinetic friction depends on the twist angle, because what we expect if we make a sliding action, and then the two monolayer graphenes suddenly slide, then they arrest again. This is exactly when the strain, which is still present in the monolayer graphene, is compensated by the kinetic friction. This is at least our interpretation. Let's finally look at a simple model. So we have here the monolayer graphene and monolayer graphene with the overlapping bilayer graphene region. And we apply an external force Fxx. And there are two counteracting forces, which is a friction force Fr and the cohesive force. So the cohesive force is there because by sliding, you open up new surface area. And this costs energy. This is very similar to this pulling of a nanotube in another nanotube by the Zettel group, where you also have this cohesive force which attracts the nanotube back into the outer nanotube. Let's assume we have an overlapping width of W, and we consider a stripe of width delta L. Now we can estimate or determine the forces by knowing the 2D elastic modulus of graphene, which is 340 Newton per meter, and the epsilon xx that we measure, we can determine that force per delta L. We can also calculate the cohesive force by the interlayer cohesive energy density times the width, again, delta L. So from that, we can derive the friction force and the difference of these forces. And in order to determine a friction coefficient, we also need the normal force, because the friction force, the friction coefficient, as we will see in a second, depends on both the friction force and the normal force. And for the normal force, we can use the adhesive pressure in bilayer graphene, which is shown here, and of course the area, which is W times delta L. So using Amonton's law, which says that kinetic friction coefficient, or also the static friction coefficient, is the ratio between the friction force and the normal force, we can now insert these formula here and get an expression for the kinetic friction coefficient. And just to remind you, we measure two quantities. So one is the epsilon xx, it's a strain in the monolayer graphene, which we use as a spring constant. And so to speak, we have here a spring constant, and we measure the extension of that spring. And we measure, of course, the width of the overlapping region, which we directly get from the image. By doing that, Xin derived this kinetic friction coefficient, which also depends now on the twist angle. And you see that the twist angle, that at a twist angle of about 1 degree, the kinetic friction goes below 0.01, which is sometimes stated as the border to kind of superlubricity regime. Unfortunately, we were not able to kind of explore twist angles smaller than 0.5 degree because already during the first sliding event, the whole system moved to these larger twist angles, probably because of this incommensurability, because as soon as you have individual dislocations, it might be energetically unfavorable to stay there, but to make another twist to reduce the friction force. So what is the outcome? With increasing twist angle, the kinetic friction coefficient decreases. At a twist angle of about 1 degree, the kinetic friction enters the superlubricant regime. And what can we do else in the next step? We would love to assess the friction behavior for very small twist angles, which is particularly interesting. So what about the transition from the lock-in situation to the first sliding situation? And then we aim for further mechanical tests combining 4D STEM to also address the static friction. So you could imagine that you also kind of slowly strain the system, and before sliding action occurs, you can already measure the straining during this period. This was not possible for these experiments, but in principle, you can independently determine static friction and kinetic friction. With that, I'm at the end. I hope that I could show you that based on our earlier investigations of dislocations in bilayer graphene, their manipulation, we now moved on to the in situ sliding of a whole network of dislocations, or in other words, on a twisted bilayer graphene, and how these dislocations evolve, and could show that a twist angle is introduced by sliding and leading to superlubricity in the sliding action. And here I want to mention the people from my group who contributed: Xin Zhou, Mingyang Wu, also technician, Carmen Rubach, and a student, Robert Kammel, and another technician, Tadahiro Yokosawa. And Colin Ofos helped us with the evaluation of 4D STEM data. So thank you very much, and I thank you for your attention.
Great, thank you very much, Erdmann. I'm not sure if you're able to see the Q&A on my phone. It's a little bit difficult to navigate there. I'm not sure I see anything here, but maybe I'll just kickstart the question. First of all, really beautiful presentation. My particular work is interested in optical properties, and these moires host really nice interlayer and intralayer excitons. Do you think it would be possible to do kind of combined cathodoluminescence either in the SEM or TEM to see how the sliding impacts the exciton energy or line width?
Yeah, so we actually started a little bit. It's very difficult, of course, to transfer the holder and the straining into if you want to do it in a separate instrument. So we started with a colleague expert in Raman spectroscopy, and she's also doing PL, but in particular Raman, and we wanted to see how the straining affects the Raman spectra locally. And this was partly successful already, but I do not yet have results to show. But the next step, of course, would be to look more to the optical properties. Yes.
Cool. Yeah, I think that'll be really exciting too, like looking at the Raman, and especially in certain classes of materials, the 2D materials might be a nice example. Some of the strain can give rise to really efficient optical nonlinearities. Yeah. So I think if you can start doing some basic characterization, who knows, there could be many years worth of really neat optical properties to disentangle.
Yeah, the challenge is, of course, to get the experiment running, because it's not something that you can 100% plan. Maybe like the cryo EM, there are always a little bit of course of luck that you have to have. And I think this has to be improved in order to make a dedicated and also systematic analysis. Okay, there's
so Jen, there's a question from Kate Reedy. Do you see it in the Q&A?
Oh yeah, perfect. So Kate is asking if you predict the superlubricity would work for other two-dimensional heterostructures or mixed dimensional heterostructures, or do you think it is mainly graphene?
No, I don't think it's mainly graphene. It's the principle should work for many different situations if you reach incommensurability. And we are currently talking to colleagues from mechanical simulation to also make simulation of the situation. But I don't think so. It's just that we are most familiar with graphene. We also started to perform experiments in MOS2. But from the experimental point of view, it's maybe even easier to do this for MOS2. And of course, if you make a heterostructure, you also may have a misfit between the lattice constant, which can help again to get incommensurate structure, because you do not have to rely only on the twist, but already the heterostructure has maybe an incommensurate relationship between the two films. This would be very interesting now to apply the same concept to heterostructure. Yes.
Also, thanks, Kate and Jen, for the questions. Also, Erdmann, your twist angles are rather small, and so we've been more interested in the larger twist angle regimes, as you know. And so what do you think expect to happen at larger twist angles, and will they be interesting?
Yeah, I mean, in principle, of course, there are, again, twist angles where you get kind of lock-in structures. So not each twist angle is, of course, incommensurate. Yeah, but these are like with grain boundaries, you have certain coincidence side lattices, and then one would expect to see an increase of friction force. But I would expect that this is very difficult to kind of reveal experimentally, these kind of plots of friction as a function of twist angle, because you cannot fully control, because the whole system is not stiff. You cannot say, I make a twist angle and just pull on it and look for the friction force, because the system reacts on your forces and may twist or may not twist depending on what is energetically favorable. And we also saw this gradient of twist angle from bottom to top, which you cannot avoid, but we could even exploit it to have more data for different twist angles.
Right, and so of course the difficulty with that is the hexagonal system does not have is irrational, and so you don't get specific coincidence side lattices.
Not specific, but close maybe.
Close, yes, I agree.
Yeah.
Yeah, that's certainly intriguing. So,
maybe just one more quick question, just trying to see if we can link the first speaker, Alexis, with your talk, Erdmann. You know, one of the, I think, benefits of the EMX symposium is that it brings together folks from the life sciences and biological community with those from the physical sciences, and there are a lot of, you know, nice cross-pollinations of ideas, especially, you know, maybe techniques that the more physical scientists, you know, pioneer that the life scientists can use. And you know, I don't want to put either of you on the spot, but I wonder if there might be the kernels of some new ideas in kind of the work you've developed to look at the kind of coefficients of friction to perhaps extend that from, you know, maybe the more model, you know, two-dimensional materials to looking at protein-protein interactions. And, you know, I can imagine that's probably pretty complicated, but yeah, I don't know if there are any simpler, you know, biological molecules or proteins where it could be neat to start looking at how, you know, molecules are sliding over, I don't know, lipid bilayers or proteins.
Probably the scanning probe techniques are there better suited to study that, because in principle you can, of course, move particles, move proteins on top of maybe other proteins or surfaces. But it's of course related, because I would expect that there are also weak forces like van der Waals forces dominate, or other weak forces dominate that.
Okay, let me try to chip in the answers to that. In the biological system, definitely they continuously moving in a living system. So there are all kinds of movement, sliding movement, skew movement, and hinge motions, and so on and so forth. And we definitely observe that from using a cryo-EM or cryo-electron tomography. And so those, some of the system, we actually have solved it at kind of atomic details in such a way we know which part of the protein is making interaction with which, and then when you start walking or sliding, and what what kind of chemical bonds has to be broken. So I think these are constantly being observed in the cryo-EM situations. But if you want to look at kind of live movement, then we turn into the optical imagings, which we can definitely see that as well. Alexis, you want some more comment?
Well, I just wanted to point out another area that might be interesting to look at is we actually look at many proteins on top of graphene derivatives, usually graphene oxides. But I think it's one of the questions that the field of cryo-EM still has to answer is how proteins interact preferentially with some surfaces and not others. So, for example, we often vary the properties of our surfaces to make the proteins behave in different ways. But these are very unpredictable experiments. We'll change surface charging or hydrophobicity of the surface and then check, does our protein interact with the surface in the same way or in a different way? And here I have a feeling generally that this is an understudied, from a physical standpoint, understudied kind of problem where we don't have predictability. We don't have a good model for understanding how proteins interact with surfaces like graphene or graphene oxide.
I'm just wondering also about the imaging here, Erdmann. I think you said there were dark field images. Are you using them in just TEM mode, or are you doing 4D stem reconstructions, or how and what voltage are you using and doses and so on?
Typically, we use 80 kV, and the images that I showed are really taken with the dark field TM mode.
Yeah.
But of course, one could also do 4D stem and reconstruct the contrast. But I think the dark field mode is maybe better to follow the dislocations directly and see what's happening. And also maybe more dose efficient if you in relation to the resolution that you can achieve. But of course, this can be performed also in 4D stem mode and virtual aperture reconstruction. That's true.
Also, it's the in situ. If you're doing in situ, then the TEM mode is still superior.
I mean, as I mentioned, the movement of the film or the sliding is too fast to capture it, even with the dark field imaging. As you can imagine, like a stick slip motion is very fast. So only if it's arrested again, we can investigate it. And I think there's also a related question now asking what is the effect of the overlap area. So if the overlap area would stay larger as in the beginning, would we expect to see different measurement, or would this affect the measurement? So I don't think so. I mean, we cannot keep the overlap area constant because we have this slip action. Therefore, I cannot completely answer it. But of course, we did not evaluate the very last slip motion because then the overlapping area is too small and maybe already some edge effects set in. So of course, the larger the overlap area, the less important is the edge, which always makes an additional effect, at least is expected to make it.
You mentioned about the sample was difficult to obtain or difficult to find, and so the sample preparation sounds pretty critical as well.
Yeah. Yeah, this is definitely the case, and Show actually used also sometimes, if we see this configuration, he used the electron beam to cut other parts of the samples to set this part free for the experiment. But and of course you have only one shot for each configuration, and you have to take all the data, and you don't know the stick slip motion in advance, whether there are many small steps of sliding or is a big step of sliding. So this makes the experiment extremely challenging.
Yeah. I think that pretty much wraps up our time, Jen.
Yep. Bob, do you want to have some closing remarks?
I just want to thank Erdmann for just a really elegant, fascinating talk, and of course Alexis as well. So maybe I'll pass it on to Wah and see if he'd like to make any remarks as well, and if there are any more questions from the audience or anyone would like to join in. I think Yi is able to let them join for the discussion. Wah?
Okay, so I like to thank Erdmann for these interesting talks. And as Alexis pointed out, we actually are pursuing a lot of graphene as a substrate. And there literature in our field that using some types of graphene treatment, one can solve so-called preferred orientation problem. As one of the audience raised a while ago to Alexis. And also their literature suggests using the properly treated graphene, one can thin a very thin ice, and not only thin, but not wrinkle ice. This is one of the things that we embedded our molecule in ice. If the substrate surface is actually important, because the ice can be kind of up and down. So you're looking at very small protein as Alice is trying to do, and then they are wrinkled, then the contrast will be minimized. So I think there's actually a lot of possibility using the graphene. I personally like to engage with Jennifer and Bob and other colleagues in material science and how we can give another push on the graphene in both fun application in material science as well as in biology. I look forward to that discussion in the near future, hopefully after the new year.
Yeah, well, we can always put some of your proteins in between the graphene as well and send it over to Erdmann to pull on it.
Yeah. Okay,
So,
I think Alexis have gone. I believe he has to take his kid to go to school. So I think if there's no further question, we may conclude today's discussions.
yes, I concur with this, and it's been fabulous. What a way to end the calendar year. Both very, very clear talks, and the goals and the application of electron microscopy is just incredible. And I'm always blown away by Erdmann's work, and I'm very impressed by Alexis also. So we have our greetings from Patricia Koyman from Cape Town. Thank you, Patricia, and for your continued support of this, and we're glad that you were able to tune in as well. So from the Northern Hemisphere, where it's cold and dark, to the Southern Hemisphere, where it's nice and warm. So we'd like to give our best wishes for the holiday season to everyone who's been attending. I'd like to thank Yi for single-handedly putting this together and helping us, and of course Alexis and Erdmann, and look forward to seeing you next time soon. So happy holidays, everyone. Take care. Bye-bye.
Bye-bye.